Bismillahirrahmanirrahim. Assalamu alaikum to all of you. I welcome all of you to third year MBBS and to the Department of Pharmacology and Therapeutics. Although our start has been a bit turbulent, but let's all be very hopeful and optimistic that about what the future holds for us. And let's be hopeful that we'll soon be taking lectures, inshallah, in the lecture theaters, and we'll soon be seeing all of you in person. Anyways, talking about the lecture and coming to the subject matter, the topic that I will be covering today, it is from the section or the unit of general pharmacology, and we have already attended to one lecture on this particular topic of general pharmacology. The three lectures that you are going to have today are all on the very basis of general pharmacology. You will be learning about what pharmacology is, what are the various branches and divisions of pharmacology, what are various active principles in the crude forms of drugs, and how different drugs are administered, the various methods of drug administration. So all these three important areas, which are actually the very basis of general pharmacology, are going to be covered today. And from tomorrow onward, inshallah, you will be starting off, or we'll be starting off with lectures on pharmacokinetics, and which will soon be followed by lectures on pharmacodynamics. So pharmacokinetics and pharmacodynamics, although I do understand that these are two entirely new terms for you, and you have little know-how of what these things are, but soon enough, inshallah, you will be having a very clear understanding and a good concept of both these terms. So even if I do use it, I'll try to explain these terms. I'll try to give you a brief introduction of these terms if I use these terms, but be assured that you will be covering both these things in a lot of detail in the days to come, inshallah. Anyways, starting off with the lecture, today's lecture by me. But let me first introduce myself. Although I've given a very brief introduction of myself along with the lecture, along with this, along with my lecture upload, you might like to skim through that, but in case you don't want to or you haven't, I'll just tell you briefly about myself that I'm Dr. Abira Sikandar, graduate of King Edward, year 2008, and then I did my post-graduation from Army Medical College in 2017, and since then I have been working here as a senior lecturer in the Department of Pharmacology, WMC. I think that's pretty much enough introduction for here, because I'm afraid we cannot have a lot of time at our disposal for more conversations. Anyways, now coming to the topic, which is sources of drugs and active principles of drugs. So I've already told you that the three lectures, and one of these three lectures is the lecture that I'm going to deliver, are on the very basis of the basic concepts of general pharmacology. I would say, these three lectures are going to introduce you to pharmacology. In this very lecture, you will be learning about what are various sources and origins of drugs, and what is the active or chemical or pharmaceutically active component which is present in various drugs, and which is responsible for the pharmaceutical activity of any drug. So we will be talking about the sources of drugs and the active principles or the biologically active substance which is present in a drug and is responsible for its activity. So moving on, we'll be, as I told you, it's actually two things combined into one. So our first three learning objectives, in which we'll enlist different sources of drugs, we'll elaborate each source with examples, and we will be talking in detail about the latest source, which is recombinant DNA technology. And in the second section, we will be talking about the second component of our lecture, which is the active principles of crude drugs, and some brief description or some brief overview of certain active principles. Now, let's move on. Let's start learning about what these two things are. So the first thing is the sources of drugs. Um, if by the end of this lecture I do succeed in stimulating this thought process in you, in which each one of you, before taking a drug, thinks about its origin, thinks about its source. Where is this drug coming from? Is it coming from an animal source or plant source? Or it's entirely manufactured in factories? It's entirely man-made, chemical? If I succeed in stimulating that particular thought process, then that's going to be a huge success for me. So, in your day to day lives, you might have some of you might be suffering from iron deficiency anemia and might be taking iron supplements for that particular ailment. And maybe some the parents of some of you might be taking some antihypertensive drug, or God forbid, some anticonvulsant or a sleep inducing drug, or maybe grandparents of some of you are on insulin. And what if someone of you falls ill, falls sick, and is given an antibiotic in a hospital setting or on OPT basis? So have you ever thought, where does this drug actually come from? Is it entirely manufactured, exclusively manufactured in a factory setting? It's totally chemical based, or does it have some natural ingredients, or does it have exclusively natural ingredients? So you should be thinking about this particular aspect of pharmacology from today onwards, inshallah. So let's start. It's just this particular slide is just enlisting certain sources of drugs, which we'll be talking about in some detail. So the sources of the drugs could be plants, it could be animals, minerals, microbes, and it could be totally synthetic, and it could be genetic engineering or recombinant DNA technology, which is the latest drug manufacturing technique available to us. So we'll be taking up all of these sources one by one. Talking about the plant source, actually, this is the oldest sources of drug. This is the oldest that is known to mankind, and it is here since very ancient times. And you might have read about it in books, and might have seen in movies or in dramas that the doctors from the ancient times, they would made these medicinal products, and they would extract certain things from the plants, and they would make them in the mortar and pestle, and they would just grind those ingredients or whatever extracts they would extract, and they would just prepare by themselves. Those drugs were prepared themselves, and the sources were mostly plants. So anyways, the thing that is important to remember, that it is one of the most, it was one of the most important source of drugs known to mankind, and it is the oldest, although not very much in use nowadays, but has been there for a large amount of time. And again, it's also important to note that almost all the parts of the plant, like leaves, flowers, seeds, roots, bark, each part of the plant can be used or has been used as a medicinal product. Not of all the plants, but certain plants which have been used, which had these properties, which had these medicine-like properties, different parts of their of those plants have been in use. Now, this slide and the slide after this is going to give you an idea of how, of what I was telling you in the previous slide. I was telling you that various parts of the plants have been in use. In certain cases, or in certain plants, for example, in this case of plant which is named as digitalis, its leaves are used to make medicines, and digoxin is that drug that is derived from this plant digitalis, and it is still very much in use, although newer drugs are replacing it swiftly, but still it is very much in use, and it is a drug that is used in chronic heart failure. Similarly, there are certain plants whose barks are used for extraction of drugs, like quinine. You might have heard about this drug. It is a drug which is used in certain cases of malaria. and it is derived from the bark of the cinchona plant. So you see that either the leaf or the bark or the fruit or the seed of various plants can be used to extract or prepare various drugs which can be used in various clinical conditions. So another example would be morphine, which is derived from the opium plant, the fruit of the opium plant, and it is an analgesic. Likewise, um, again, that's a more of a repetition of of previous slide. So let's move on. Here we are. Another good example is another drug, which is actually a group of drugs called vinca alkaloids, and these are very potent anti-cancer drugs, and they are also derived from plants. So you can see, and you might have now understood this fact that plants have been various parts of various plants have been a very good source of various drugs, and it is known to mankind since a long time now. Now, another important source of drugs other than plants is the animal source, and it is also in use since a long time, and it is also um, is of course it's organic, and it's again, as I told you, that various parts of the plants could be used for preparing various medicines. Similarly, various tissues and organs of different animals can be used to produce different drugs. Giving you some examples is that pancreas of certain animals, like that of cow, can be used to extract insulin, and that insulin has been administered for human use, or was in human use for a long time. Although now that insulin is now replaced by human insulin via the recombinant DNA technology, but for a long time, the insulin that was derived from the pancreas of certain animals was used for the patients of diabetes. Similarly, the cod liver oil, which contains vitamin A and D, is a very good example of drugs from animal sources. Again, giving you a list of some drugs and their animal sources. Insulin, as I've already mentioned, that it is coming from the pancreas of cows. Protamine sulfate, you would learn soon that it is an antidote to a drug used in blood disorders. It is an anticoagulant, and this is being derived from fish. Pig's intestine is a source of heparin, and heparin is an anticoagulant, and heparin is also derived from ox. So various animals giving us various drugs. Again, pancreas of cow, pancreas of pork giving us insulin, stomach of cow is giving us pepsin, which is a digestive hormone, and although not very much in use, but because of some newer drugs, but was very much a mainstay of treatment in older days. Thyroid hormone, people who are hypothyroid need to take thyroxine exogenously. And this thyroid was or is even now produced or is derived from thyroid glands of certain animals. So now you know that animals and plants are two very important, although old, but very important sources of drugs, which are very much used even today. Now, another important source, or a third source of drugs um, uh, which are in use, in human use, are minerals. Minerals or earth or surface of earth. For example, in the beginning of the lecture, I gave you an example that if someone of you is anemic and is taking iron supplements, you might like to think about the source of that iron. That iron is of course is an elemental iron, and is of course coming from is an earth element. It's coming from the surface of earth, or in certain cases, I think it is the earth. It's coming from iron cores. Then there are certain other elements like magnesium sulfate, which has some gastrointestinal uses. Then there are some other minerals like sodium bicarbonate, which are being used as antacids, aluminum hydroxide. So all of these drugs are very much a component of various pharmaceutical preparations, particularly preparations which are used for gastrointestinal effects like antacids or purgatives. And they are all derived from natural, or you can say metallic or non-metallic sources coming from our earth. Okay, sorry, I have to move forward. So some more examples. Zinc is a very good example of these of drugs which are coming from mineral sources. Um, this zinc is very much a component of some sunscreens and some rash creams, and it has a very good healing properties. Then gold has been a very important component in the treatment of this of disease of rheumatoid arthritis, although it has been replaced by newer drugs, but has but still is a second line drug for the treatment of rheumatoid arthritis. So far we have learned that animals, plants, and minerals are some of the very important natural sources or origins of various drugs, which are very much in use even today. Okay, moving on to another natural source of drugs, which is that of microorganisms. You might have heard that story of discovery of penicillin, and which was discovered in 1928 by Scottish scientist Alexander Fleming, and it was an accidental discovery. He was actually growing some bacteria in his laboratory, and that bacteria, when a mold of Penicillium notatum grew over that bacterial culture, that bacteria that it began to die. So this is how the this was the first antibiotic to be discovered, and this is how the era of antibiotic began. So a large number of antibiotics, in use, very much in use even today, like penicillins, chloramphenicol, cryzophyllin, streptomycin, neomycin. I am very much aware, my dear students, that these names are new and very difficult for you to understand and pronounce, but it's only you just give it some time, and inshallah you will just ace it. So the message for today is, or the important thing that you need to learn today is that the drugs are coming from various sources. Not all the drugs are being exclusively manufactured in the factories or in the industries. They are also coming from the natural sources, and some of these natural sources are then chemically altered to produce altered form of chemicals in the factories, and that particular um, form of, or that particular source would be called semi-synthetic derivatives of drug preparations. So actually you can say that the drugs are either totally naturally produced or totally have a natural source, or sometimes they can have exclusively synthetic source, or sometimes it is a mixture of two. The natural product can be altered chemically to give us a semi-synthetic source. Animal source, plant source, microorganisms, and mineral source is exclusively natural. And in this particular slide, you are being shown a list of antibiotics, which is coming from certain microorganisms. And for example, Penicillium notatum, Penicillin cryzophyllin, streptomyces, these are all fungi, and these fungal organisms are a source of various antibiotics. Okay, again, my computer loves to go back. So after having covered the natural sources, let's talk about the synthetic sources. A large number of drugs that are in use nowadays are synthetic. And when the nucleus of the drug from a natural source is altered or is changed, as well as the chemical structure which is attached to it is altered or is changed, it is called synthetic. So both the things are being changed. The nucleus of the drug from the natural source, as well as the chemical structure of it is being changed in the factory or in the industry, then that is called the synthetic source of a drug. Okay, so of course these synthetic drugs have a large number of advantages. For example, they are pure, you can be very much easily control their quality, and they're more potent and they're safer, unlike the drugs which are derived from natural sources and are given directly for human use purpose, for humanly use. And of course, because they are synthesized in the laboratory, so large scale production is very convenient. Here is giving you certain examples. So by and large, most of the drugs that you will be studying throughout the year in pharmacology would be synthetic. Some would be natural and some would be semi-synthetic, but most of the drugs will have their source as exclusively synthetic. Now, the third source is semi-synthetic. And in this case, the difference from synthetic is that the nucleus of the drug obtained from the natural source is not changed. Remember, in case of synthetic drugs, the nucleus of the drug as well as its chemical structure both were being changed. But in the semi-synthetic source, the nucleus of the drug obtained from the natural source is not changed. Only its chemical structure is altered. And that is when we call it semi-synthetic. Okay, some examples are atropine is a natural drug, is a natural derivative. And if you change some of its chemistry and do not change its core nucleus and just change a little bit of its chemical structure, for example, by adding bromide to it, it becomes atropine bromide. Then similarly, you can again do some changes with its chemistry and make it homoatropine. Similarly, ampicillin can be produced or semi-synthetically produced from penicillin. Ethinyl estradiol can be produced from estradiol. Methyl testosterone can be produced from testosterone. So all these are the examples of semi-synthetic drugs, and they are just being produced by some minor alteration in the chemical structure, and the core nucleus is not being changed. So it's pretty much repetition of what I've already told you. Then there are certain drugs nowadays, ever since this recombinant human technology is very much in use, and we will be talking about this in detail in the coming slides. But some hormones, especially the regular insulin or growth hormone or menotropins and human chorionic gonadotropins, they are being extracted from humans and are, of course, after recombinant DNA technology, are being administered for various ailments or diseases in humans. So before we formally begin talking about the recombinant DNA technology, we'll be talking about this particular source of drug production in some detail. Let's just quickly recap that drugs are not just exclusively manufactured in the factories, but some of the drugs are being produced or are being derived from animal sources, plant sources, microorganisms, and in certain cases, the source can be mineral sources or human sources as well. But sometimes if you just change the chemistry, you just give some minor alteration in the chemistry, the source would become semi-synthetic source. And if you change the chemical structure as well as the core nucleus of the substance, which is derived naturally, then you are going to have a synthetic drug. Synthetic drugs are by and large, most of the drugs that you are going to study in pharmacology are synthetic drugs, but a large number or a lot of drugs you will soon be knowing this are exclusively coming from natural sources as well. So talking about recombinant DNA technology, what is it and why is it so important to learn about it? This particular section of this lecture is really important because you might get to write a note on it, or you might be asked about this in Viva, and particularly what this is and what is its application in pharmacology and in medicine. How has this revolutionized drug therapy and treatment of various diseases. So recombinant DNA technology, basically, although I'm guessing and I'm assuming that you have learned about it in biochemistry in your previous years, but even if you haven't, or um, even if you have or haven't, doesn't matter. We will be talking about it in detail in this lecture as well. So it is something that involves cleavage of DNA by enzyme restriction endonucleases, and you just select and identify a gene of desire, and you eventually couple it to the plasmid of a rapidly replicating organism, and then eventually you get a recombined DNA, or a new genetic combination is formed, which is eventually inserted into a bacterial culture, and it allows production of a vast amount of the genetic material. For example, if you consider the example of insulin, if you have to produce insulin from an animal source or from a human source, and you have to produce, or you have to have a large scale production of insulin for um, uh, for use, what should I say, for use at a larger scale, what do? You restore to this particular technology. And I've told you what this is about. And this figure is pretty much summarizing everything that we will be talking about in the slides to come. We'll get to this slide later. But first, let's talk about what plasmids are. Plasmids are extra chromosomal circular rings that are separate from the cell's own original chromosomal DNA. And again, recombinant DNA technology is recombined. Okay, just leave these definitions. First, let's get to this definition. I really like this definition, so I have incorporated it here. It is a technique which is used in genetic engineering, and it involves the identification, isolation, and insertion of a gene of interest. For example, gene of insulin for the treatment of patients with diabetes, that is identified, isolated, and inserted into a vector. And vector is mostly a plasmid, and we've just talked about what a plasmid is, such as a plasmid or a bacteriophage, to form a recombinant DNA molecule and production of large quantities of that particular gene, for example, the gene fragment for insulin, for example, or the product of that is not just the insulin gene, but eventually the product, that is insulin, which is encoded by that gene. So if you just are asked about the definition, you can always write this one or the one which is mentioned elsewhere in the lectures, or you can look up for your own definition. But the definition is really important, and knowing the various steps of recombinant DNA technology is very important. So talking about the various steps, this particular slide is giving us an idea of what various steps are, and we'll just briefly talk about these. For those of you who are interested in knowing further details of this recombinant DNA technology, I've also uploaded or attached a very small 3 to 4 minute video from YouTube alongside the lecture. And those of you who are interested and would like to know more on this topic can always go and watch that video. But knowing this much would also be sufficient. So the step one is identification and isolation of gene of interest, or a DNA fragment that is to be cloned. Step two is insertion of the isolated gene into a suitable vector, which is the plasmid. Then step three is the introduction of this vector into a suitable organism. This process is called transformation. And then eventually selection of the transformed host cell. We'll talk about what that is. Multiplication or expression of the introduced gene into the host. So what recombinant DNA technology is, and why is it so important? It is so important because it has enabled us human beings to extract or to produce large scale production of certain natural elements, like natural hormones like insulin, like growth hormone, like erythropoietins, like interleukins, and that has only been possible because of this recombinant DNA technology. And it has many other applications other than its pharmaceutical or medical applications. Anyways, coming back to the various steps involved in this. The first step would be, of course, that you have to identify, or you have to locate, or you have to uh, isolate the gene of interest. For example, if you want to produce insulin on a large scale level, you will identify, you will isolate it, for example, from a genomic library. Okay, now you have isolated the gene of interest. From where you can get this gene of interest? You can get it from genomic library, or you can even chemically synthesize it in the laboratory if you know the sequence. Anyways, once you have isolated gene of interest, the second step would be that you add this gene of interest to a plasmid. And is this plasmid is, as I've already told you, is an extra chromosomal ring which is capable of replicating independently from its you can say from its from the chromosome of the organism that it is belonging to. And what we do is that we just cut out a piece of this a piece of this DNA by the help of certain restriction enzymes, and with the help of certain DNA ligase enzymes, the gene of interest is incorporated into this plasmid. So when the plasmid is combined with the gene of interest, the thing that is formed is called the recombined DNA, or the recombinant DNA. So a vector is anything, any DNA molecule that has the ability to replicate inside the host to which the desired gene has integrated for cloning, and we'll be seeing how it works. So step 3 would be that once the recombinant DNA with the plasmid, with the gene of interest being incorporated into the plasmid, is eventually incorporated or is introduced into a bacterial culture. Okay, mostly the bacterial culture is that of E. coli. Now this E. coli has this gene of interest incorporated in its structure, and this process in which the gene of interest is eventually incorporated into a bacterial culture, mostly that of E. coli, is called transformation. Now what next? Eventually, you put those E. coli, that culture, and you allow it to grow in a selective medium. And once it is allowed to grow, you will see that it is going to be multiplying in various colonies. Some colonies are not going to have any plasmid in it. Some are going to have the plasmid, but it is going to be unaltered. And some colonies will have that thing which we desire. That is the plasmid along with the gene of desire. So you are going to select that particular colony, and in step 5, you're going to allow that particular colony to multiply and eventually to give you a large number of copies of the gene of interest. So you, uh, you can always describe this process of recombinant DNA technology or genetic engineering in 4 to 5 steps. In identification, isolation of the gene of interest, incorporating that gene of interest into the plasmid vector, transforming that plasmid vector into a bacterial culture, allowing it to grow, and then selecting the desired colony and allowing that colony to multiply so that you get a large number of copies of the gene of interest. And this is how this recombinant DNA technology works. In case some of you want to know more about it, there are some more slides if you want to go through those. But whatever I have explained in those 4 to 5 slides is sufficient. So this particular figure or schematic representation is giving you very much an idea of how this recombinant technology is working, how these gene of interest, after being incorporated in the plasmid and we are allowing it to replicate, and eventually this has so many applications at so many levels. For example, It can be used to dissolve blood clots in heart attack therapy. So this is, we're talking about a drug which alteplase, and we'll talk about this drug later in the slides to come. So we are actually, with the help of this particular technology, we are being able to produce drugs. We are being able to produce crops which are genetically modified and resistant to so many um, insect invasions, and they don't need a large number of insecticides and pesticides for their good growth. So this is about briefly about the recombinant technology. So the important thing so far that you have learned is that there are so many sources of drugs. Some of them are natural, some are synthetic, semi-synthetic, and amongst the synthetic source, recombinant DNA technology is one of the most recent and the most relevant technology that is in use, and has so many applications at the agricultural level, as you can see, as it's written over here. And as it has so many, it has given us the opportunity to produce so many vaccines, and is allowing us to do gene therapy and antiviral therapies, and also some very important hormones which are used for some very important, again, which are used in some very important diseases like diabetes and dwarfism, are being produced only because of this technology. It's a very interesting figure, and that is why I've added it for you, so that you can see how this particular technology helps us in producing vaccines, how a harmless and a harmful DNA can be combined together and eventually can be injected into the human body, and we can have a human immune response. And this is how vaccination can be attained with the help of recombinant DNA technology. This is a very important slide. It is telling you, it is enlisting a large number of very useful drugs and hormones and growth factors that are being produced because of this very important technology. So just summarizing it, you need to know its definition, what a vector is, what are the various steps involved, what are its applications, and mainly its applications in in the pharmaceutical industry and in the production of various medicines. So here's a list of those medicines that are being used against various diseases. And I would recommend that you do go through that particular slide really well. Okay, now coming to the second section of our lecture, which is the active principles of crude drugs. So far we have learned about the sources of drugs: plants, animals, recombinant DNA technology, synthetic, semi-synthetic, and all that. Now we'll talk about what are the various active principles which are present in the crude drugs. The crude drugs, by crude I means which are totally natural, and they are not being, you can say, they are not being modified or purified. They are absolutely natural, and whatever chemical constituent is present in that crude form and is responsible for the biological activity of that drug is called the active principle. I hope it is clear. So the active principle is the chemical substance which is responsible for the biological activity of any drug. Here's enlisting the active principles. You don't need to get into the detail of each one of these active principles. Out of all these, alkaloids, glycosides, fixed and volatile oils are the most important ones. But it's better to have an overview of all of them and to have a concept of all of these with examples. Anyway, starting off with the most important one, which is alkaloids. And alkaloids, as the name indicates, anything which is an alkali like is called an alkaloid. And amongst the most important natural active principles, this is going to top the list. So it's very active. Of course, it is basic, and it is a very complex natural nitrogenous compound, which is it is found only in plants. So the chemical constituent of those drugs which are extracted from plants and are nitrogenous in nature and are very bitter in taste are called alkaloids. So you might be asked about what an alkaloid is. So how will you answer this question? Alkaloid is an active principle of the drugs which are extracted from plants, and they have a profoundly bitter taste, and they have a complex nitrogenous structure, and they are alkali-like chemically. And when we say something is alkali-like, means that it reacts with acid to form salt and water. And another important thing about these alkaloids is that generally they have this suffix ine, -ine, with their names. All their names, they end with an E. For example, atropine, morphine, quinine, nicotine, strychnine, rezapine, pilocarpine. So all these drugs are actually derived from plants and are alkaloid in nature. So what is the source of atropine? Source of atropine is a plant. It is an atropine belladonna, and the nature of the active principle of atropine is an alkaloid nature. Similarly, nicotine, it's also derived naturally from a plant, and it also, the nature of the active principle of nicotine is alkaloid. Similarly, quinine is an alkaloid, morphine is an alkaloid. So when I say alkaloid, you should immediately reach to the conclusion that an alkaloid is a drug which is produced from the plant and is very bitter in taste and has a complex nitrogenous structure, and chemically it is an alkali-like substance. Some examples of alkaloids, we have already listed them, and there's another list. You might like to go through this. Atropine, hyoscine, hyoscyamine, quinine, quinidine, cinchonine, cinchonidine. You don't need to remember all these names. You will eventually learn these. You will have an acquaintance with these names because they are going to be repeated over and over again. So don't worry about the names right now. So these are some plants, and they are giving us the active substance, which is alkaloid. Example, morphine as an alkaloid coming from the plant opium poppy. Quinine is an alkaloid coming from a plant, from the bark of cinchona plant. Atropine is an alkaloid coming from the fruit of the plant Atropa belladonna. Cocaine is coming from the plant, is an alkaloid coming from erythroxylon coca, and so on and so forth. Glycoside is another natural active principle, and this is also coming from plants. Glycosides has basically two components, a glucose component called the glycon component, and then a glycon component. The a glycon component is responsible for the pharmacodynamic properties, and the glucose component is responsible for the pharmacokinetic properties of that drug. The most common example, or I would say the only example of this particular group, is that of digitoxin or digoxin, and it is coming from the leaves of foxglove leaves. It is coming from this plant, foxglove leaves, and this drug is very much has been the mainstay of treatment of uh, chronic cardiac failure. And what else about glycosides? They are also very active. And again, this I've already told you this fact that the non-sugar component is the active component, is the pharmacodynamically active component. And again, their name, as in the case of alkaloids, their name, however, ends with an IN. In that case, the name would end with INE, like atropine and pilocarpine and quinine. In this case, it is digoxin, digitoxin, gytoxin, and ouabin. So far we have learned about two principles. This is giving you a structure, an overview of the structure of digoxin. We will learn about this structure when we talk about this cardiac glycoside in detail in cardiovascular pharmacology. Then we have another natural active principle of drugs, of crude drugs, and it is saponins, very much like glycosides, again coming from plants, and is an emulsifying agent. Um, and I think this is pretty much enough for this, and you may just like to learn one example, and also that it is very toxic, generally not used as a medicinal product. Now, this is an important area of this particular section of my lecture. What are the various types of oils which are the active principles of various drugs? One type of oil is the volatile oils, which is also called the essential oils or the ethereal oils, and the second part is the fixed oils. Volatile oils are obtained from various parts of the plants, from all the parts of the plants, I would say, except for the seeds, and they're obtained by distillation, are highly aromatic, and are mostly liquid hydrocarbons. What else about these? Slightly water soluble, can impart their smell and taste to water, generally colorless, but they can turn rancid and they can turn dark. And that is why they have to be stored in amber colored glass bottles in cool and dry places. Now, some examples of volatile oils: oil of sandalwood, clove oil, sorry, cardamom oil, balsam of tolu, eucalypt oil. So all these are volatile oils. Volatile, as the name indicates, they are going to evaporate at room temperature, obtained after distillation, and are highly aromatic and are very reactive. So these are volatile oils. And now the second type of oils, which is the active principle of certain drugs, is the fixed oil. And they are actually esters of higher fatty acids. Unlike the volatile oils, they cannot be distilled without being decomposed. And they form soaps with alkali because I've told you they're like esters of higher fatty acids. And your biochemistry knowledge must be telling you all this, that whatever is written over here. And it leaves greasy mark on paper. Mostly fixed oils are derived from the seeds of plants. Volatile oil is coming from parts of the plant other than seeds, but fixed oils are coming mostly from the seeds of plants like olive oil and castor oil. Here is this table that is giving you a comparison of fixed and volatile oils. I would suggest that you do go through this one uh, today, as well as whenever you are going to have an assessment on this topic. It's an important topic just to give you a concept of what these two oils are, which one is volatile and which is greasy, which is non-greasy, which is getting rancid with time, and which examples of both is oil: castor oil, cod liver oil, camphor, and methanol, and all that. So it's a very good table. You do go through this table um, today, as well as once again when you are supposed to prepare for any test. Then we talk about resins, polymers of volatile oils. Just learn one example of it, benzoin, and that's it. Oleoresins. Oleoresins are dissolved in volatile oils. Gum resins and balsams are various types. You just have an overview of these slides, of this slide and the slides that are coming after this, with examples. That's it. Gums. Gum is something that you might have seen in just exudating from certain plants. And as you know, that this is very much carbohydrate in nature, cellulose. It's amorphous, transparent, and its medicinal use is that it produces gum acacia and gum tragacanth. And this is very much used as an emulsifying agent in preparation of certain suspension and emulsion, which are used in various skin ointments and skin creams. Then we have tannins. Tannins are non-nitrogen substances, and a very good example is that of astringents. And what astringents are, you will be learning about this in another lecture on drugs used for um, in dermatological preparations. But for now, I would like you to know that astringents are substances with profound drying properties and are used as antiseptics and applied on skin as antiseptics. And chemically, the active substance in astringents is tannins. Then there are some more neutral principles which are not necessarily belonging to any special group. And an example would be that of santonin, which is used in anti-helminthic drug, which helps to propel or expel parasitic worms from the body. Then fats. Fats, as you know, are fixed oils and are being derived from plants and animals. I'm sorry for this double S. Natural esters of glycerols and fatty acids. We know what triglycerides are and what fixed oils are. You might like to remember some examples of fats: pure broma and wool fat, Waxes are esters of fatty acids with monohydric alcohols, and a very good example is that of white beeswax. Again, these waxes are used in ointments and other topical preparations used on skin. Then we'll talk about balsams, combination of resins with benzoic acid and cinnamic acid, and applied on small superficial wounds and abrasions. So, my dear students, I would like you to, about this second section of my lecture, I would like you to learn in greater detail uh, about alkaloids, glycosides, and fixed and volatile oils. And of course, you should be knowing the names of other active substances as well. But the detail or the deeper knowledge is required on those four active principles at least. So I think it was an easy topic. It was a very easy topic, I would say. And just go through it once again and get acquainted with the names, with the new names, with the names of the drugs, with the names of the active principles, and what an alkaloid is, and especially learn about recombinant DNA technology and what are its various applications in the pharmaceutical industry. So that is it. Learn about recombinant DNA technology, uh, uh, prepare it well, and prepare alkaloids well. And these are the two most important things from this lecture. And okay, thank you very much for your patient listening. And I'll be leaving you with an assignment as well. And you will be marked on the submission of that assignment. Thank you very much, and Allah Hafiz.